Okay, so I was asked a question to cover some of these questions which students are struggling with. So let's start with this one. So Divine, if you are here, then great. If not, this is for you, so you can watch back later. So when you look at this question, the question gives you a lot of information. So they're telling you about this naked mole rat. They're telling you that it's a mammal. So first of all, when I answer this question, I would always be highlighting some of the key parts that they've told me. So they've told me that it's a mammal. They've told me that there are several features that are unusual for the mammals. And this is that an unusual characteristic is that they have this tolerance of pain. So acid will cause pain responses in most mammals. However, the naked mole rats are tolerant of the pain caused by the acid. So first of all, we're talking about response. We obviously know that the acid is the stimulus, and then the response will be coordinated by the system. So we need to think about, well, what goes after the stimulus? So the stimulus needs to be detected by a receptor. That receptor will then pass the information on to the neurons, and those neurons are going to be passing that information to the coordination center, and the coordination center is going to pass that information on through the neurons again until we get to the effectors, which are going to carry out the response. So it's important that we're thinking about this, right? We've got a stimulus, which is acid. We've got a receptor that's going to detect that presence of the acid. Neurons are going to be passing the information on. So there are three you need to know about. You've got your sensory, you've got your relay, and you also have your motor. So you have those three neurons, and this tolerance can be explained by the type of pain receptor found in the naked mole rat. So they've already told us that we're looking at a receptor. So all I'm doing is I'm activating the information from my brain, pulling it forward to help me. So let's see what they've told us. So figure 2.2 shows a representation of some ion channels present in the pain receptors. And these are going to be present in both the naked mole rat and also other mammals. So the first thing we need to look at is, what are they telling us? We have acid sensing sodium channels, and this channel opens in the presence of sodium. So, excuse me, what they're going to be looking for me to think about is, what do I already know? Well, I know that cells are going to be at a resting potential, where they're going to be pumping ions in particular ways to keep this balance. So what we want to ensure is that we have the correct movement of substances from one place to another. So if we were to look then at the resting potential of a membrane, we would therefore want to bring in I'm just going to bring in some notes here. Information, which is your resting potential is your potential difference across the plasma membrane. So we know that this is the inside of our cell. We know that this is the outside of the cell. And what we want to ensure is that there is a potential difference of minus 70 millivolts. Now, to maintain that potential difference of minus 70 millivolts, what you want is for 3 sodium to be pumped out and 2 potassium to be pumped in. Now you can also see that I've got some channels here in my membrane, and those channels can be open or they can be closed. So it's important to note, for our resting potential to be maintained at minus 70 millivolts, what we want to ensure is that we have an electrical chemical gradient between the two. So we want to ensure that there is a difference between those two areas. Now, those two areas, to ensure that there is a difference between them, we have not only the potassium being pumped in, but we also have some leakage of our potassium out. And it's important to note, why are they moving out? And it's all to do with the fact that we want to ensure that we are pumping the potassium in so that we have a high potassium concentration inside and a low potassium concentration outside. So the potassium will diffuse down its electrical chemical gradient. And by pumping our sodium out, what we've therefore created is a low concentration of sodium inside compared to outside. So if we open the channel, what's going to happen is the potassium is going to move out, the sodium would move in, going down its electrical chemical gradient. So what we want to have is that our membrane potential is maintained at that resting state, which is all to do with this electrical chemical gradient. So our concentration gradient of ions is what we refer to as our electrical chemical gradient. So that's what we want to maintain. And by maintaining that, that therefore means that the ions will move in a very specific way. So let's go back to the question and just to have a look at what it's telling us. So if we look at the question, they've told us that we have some voltage-gated, change colors, voltage-gated sodium channels. So these voltage-gated sodium channels are not going to open unless we have reached the correct voltage. So what we get is then a movement of the ions either in or out if the channel is open, but if the channel is not open, then there is no movement. So therefore, if we open some channels, we can move away from that resting potential. But if we don't reach the right voltage, if we don't reach the threshold value, then these sodium ion channels are not going to open. So what you get is that your resting state is minus 70 millivolts, and only once the threshold value has been reached, because we're talking about receptors, if the generator potential does not reach the threshold value, then the voltage-gated channels will remain closed, and the membrane will be brought back to that resting state by the sodium-potassium pump. So that's really important that we keep that in our mind. Our resting state is minus 70 millivolts, or if they give you a graph that tells you something different, use the number in the graph. That's where most students fall down. They've memorized minus 70 millivolts, and actually, when it comes to the real exam, what they've done is they've given you a question that's maybe minus 65 or minus 60. And what happens is students get so hung up on that memorizing of the content, they lose the marking point because they haven't looked at the graph that's been given in front of them. That is a module 1 assessment where you are analyzing data that's in front of you. So back to the question though, they've told us that we have these voltage-gated sodium channels. Now these voltage-gated sodium channels are there. This channel is prevented from opening if there is a presence of these hydrogen ions. So if there is a presence of hydrogen ions, those voltage-gated sodium ion channels are going to not be able to open. Let's look at what they've told us in the other mammals. So again, we have acid-sensing sodium ion channels, exactly the same. They will open in the presence of sodium. So what are we doing? We are putting these acid that's going to cause a pain response in other mammals. So if there's acid present, these sodium ion channels will open. Which direction will the sodium move? Well, if we then go back to our knowledge to do with the sodium potassium pump, and we go back to our knowledge to do with the neural communication, we then should know that if we actually get up to that threshold value, if the generated potential reaches the threshold value, this will trigger what's known as an action potential. Now, that action potential is where we're going to open our sodium channels, our voltage-gated sodium channels, and then sodium will flood into the axon. Because if you remember before, we had our voltage-gated sodium channels, we have our high sodium on the outside and our low sodium on the inside. Therefore, if we actually open some voltage-gated channels, the sodium will therefore flood into the axon. So we're then having a positively charged ion move into the axon, and because it's moving into the axon, that is therefore depolarization happening. We are depolarizing the membrane. Now, a lot of students don't realize that this point here, the minute you are moving away from minus 70 millivolts, you are depolarizing the membrane. What's important is that you will only have an action potential be triggered if that threshold value is reached. So if you reach the threshold value, then depolarization, then we have our repolarization, then we have our hyperpolarization. So depolarization, the voltage-gated sodium channels open. Repolarization, our voltage-gated potassium channels open, and potassium is going to flood out. And then our hyperpolarization, we go way past the minus 70 millivolts. Therefore, we are hyperpolarizing the membrane. So going back to the question then, it says in the question that when you have your voltage-gated sodium channels, they are not affected in mammals by the hydrogen ion presence. So therefore, when it says use the information in figure 2.2 to suggest why naked mole rats do not feel pain when exposed to the acid, they cannot have an action potential because those voltage-gated sodium channels are going to be prevented from opening. So what we can see in this case is we have that there is no action potential because they've told us our voltage gated, I'm abbreviating here, but do not abbreviate in the exam, our voltage gated sodium channels are prevented from opening. Therefore, make sure you give more information. There is less depolarization of the cell, receptor cell, and that is because fewer sodium, I'm going to move it up here, it's a voltage-gated sodium, so therefore we know we had a high concentration outside, a low concentration inside, therefore we have less sodium moving in. We still have a high concentration outside, a low concentration inside. We would want it to move in, but because they have been prevented from moving in, that is what stops it from actually working. And then the second question on that one for you was explain how a pain receptor is an example of a transducer. What they're wanting you to understand on this one is they want you to understand about receptor cells. So for this one, what they're expecting you to understand is that sensory receptors are differentiated cells that are going to detect the changes in the environment, the stimuli. So if we're looking at that, we have specific cells to detect specific stimulus or stimuli, plural. So, oh, let's try that again. Went a bit too fast there. So therefore, if we have a photoreceptor, that's going to detect the light. Light is our stimulus. Our photoreceptor is our type of sensory receptor. A chemical, we have a chemoreceptor. For example, our carotid body, we have our chemoreceptors there. We have them in our aortic arch as well. Our heat receptors, they are thermoreceptors. They're found, for example, in your skin or in your hypothalamus if we were looking at the blood temperature. You have mechanoreceptors. Now, mechanoreceptors are ones which are going to detect pressure and movement. So it could be, for example, the Pacinian corpuscle, which you need to know about, or it could be a baroreceptor, which is to do with your blood pressure. So a baroreceptor in your carotid artery, a baroreceptor in your aortic arch is going to detect the blood pressure that is being pumped around the body and therefore signaling whether the sinoatrial node needs to increase its frequency or decrease its frequency. So all of these are sensory receptors. And the question was about how can we actually explain how that pain receptor is an example of a transducer? Well, all of these, whether it's light, heat, pressure, movement, chemicals, whatever that stimulus is, they are going to convert that stimulus into a nerve impulse, that generator potential. So a receptor's job is to sense that whatever that stimulus is and convert that stimulus into a generator potential, a nerve impulse. That is what a transducer is. So therefore, in the case of the pain receptor, we go back to this original one, that pain receptor, that is going to detect the presence of the acid, that's the stimulus, and that is then going to convert it into an action potential, an electrical impulse. So I hope that helps.